Hello, my dear students. We're going to continue now with Unit 2 for the AP course. And in Unit 2, we're going to dig into the cells and how the structures of cells are important and what are the components of the cells that makes all these cells function in the way they do. So now we're going into biology and pure biology so that we can understand how our cells are working and how our whole body is working accordingly. We're going to watch a short video that would make an overview of the cell structure, which is the unit that we're going to explain in Unit 2 about the cells and the cell membrane and how the organelles that are involved in all the functions of the cell. We will start with the cell membrane, as you can see. We have the cell membrane that encloses all the organelles inside. We have the cytoplasm and we have the nucleus that has our DNA. DNA, of course, is the genetic material. DNA as well is found in the prokaryotic cells and in the eukaryotic cells. We will start our video with the eukaryotic cells and starting with the nucleus because it's the most important organelle. Now, of course, when we talk about eukaryotic cells, we're talking about plants and we're talking about animals. Of course, we have the fungus and the protists as well, but the prokaryotes are totally different because they do not have membrane-bound organelles, and they have the genetic material suspended inside the cytoplasm of the cell. As you can see, it's smaller, and it has a circular DNA, and the most important example of prokaryotes are the bacteria, of course. By looking at the organelles that are found in the eukaryotic cell, we can see how the nucleus, which is the most important part, encloses inside it the DNA, which is the genetic material, of course. Now we're inside the nucleus. The DNA, as you can see, is the double helix molecule. It's a thread-like structure. And it is enclosed inside the chromatin because the chromatin is DNA and as well proteins. As you can see, these are the threads of DNA that can be coiled and supercoiled to form the chromosomes whenever we have a cell division. This is the cluster that is called nucleolus, and it is responsible for the ribosome production. And the ribosomes are important in the protein synthesis process. These ribosomes can be released through the pores of the nuclear envelope, and they will stick themselves onto the endoplasmic reticulum, where they are forming the process of protein synthesis. These enclosed membranes, as you can see, are basically the rough endoplasmic reticulum, where all the protein process is taking place. As you can see, we have some suspended ribosomes in the cytoplasm as well, but eventually they will stick themselves to the rough endoplasmic reticulum. So the RAR now is responsible for the protein synthesis, and we have the smooth endoplasmic reticulum, which is responsible for other types of lipid production. Now, this is a protein that is produced from the rough endoplasmic reticulum, which will be now in the primary structure, as you can see. It will be enclosed inside a vesicle, where it will be released out of the endoplasmic reticulum into the Golgi apparatus, where now specific things are taking place inside. Now, inside the Golgi apparatus, these proteins are going to be folded, as you can see, and they might as well be now modified. And the modification means that we might add, for example, a lipid molecule on the protein to make it, for example, cholesterol, or we might add carbohydrate to make it glycoprotein. This is basically another organelle found in plant cells, which is the vacuole. The central vacuole, which is the permanent vacuole that is found in plant cells, is much larger than the ones found in the eukaryotic cells, like animal cells. In the animal cells, we have lysosomes that is not found in plant cells, and they are responsible for getting rid of all the wastes that are not needed inside the cell by the hydrolytic enzymes, as you can see. This is the mitochondria, and it is responsible for the production of energy, which is ATP molecules. We have an internal crista membrane. This folded membrane is responsible for all the ATP production that you can see. The cellular respiration, of course, is the process by which the mitochondria is producing energy from. These are the cytoskeleton, which are microtubules that are causing all the structure of the cell to be held in place, because you know that the animal cell does not have a cell wall. So we have the cytoskeleton to hold the cells in place. The last organelle that we're going to discuss now is found only in plant cells. And this organelle is going to be the chloroplast, because chloroplasts are responsible for the photosynthesis. Chloroplasts have the grana inside them, which are stacks of thylakoid membrane, where the chlorophyll molecule is found. And of course, we have the cell wall, which is also distinguished in plant cells and not in animal cells, that is keeping all the organelles of the plant enclosed. So let's start now with the subcellular components, and we can now highlight the word cells and the cytology or the cytologists. When we say the word cytology, and logy means also always the science, it's basically the science that studies the cells. And the cytologists are the persons who are working on that science, or let's say the scientists that are digging into the cells and how they are working and what are the components of these cells. Of course, cells are the main components or the basic units for all living organisms. Now, we will talk about two different types of cells, and the first type is the prokaryotic cell. Pro means two words. And when I say karyotic, that means we're talking about a cell that is going towards having a nucleus, but it doesn't have a nucleus. So what makes it very important to study the prokaryotes is because the main cells that are prokaryotes are the bacteria. And the bacteria is an essential living organism that is affecting our lives. And that's why we need to study those types of cells. So when we talk about the prokaryotes, we're talking about the eubacteria and the archaeobacteria, which are the two types of bacteria that we were going to discuss when we reach the taxonomy unit. So by looking at the structure of the prokaryotic cell in general, we can see that this is a sample of a bacteria. A sample of bacteria may consist of the cell wall, which is the outer part of the bacteria. And then we have the cell membrane just beneath it, and the cell membrane is responsible for the exchange of nutrients in and out, or the wastes as well. And then we have the DNA of the bacteria, which is considered as a circular DNA. It's unlike the linear DNA that we have as, let's say, humans or animals in general. Then we have the ribosomes, and the ribosomes are important for the protein synthesis because bacteria, just like us, requires proteins to be produced, like, for example, enzymes or other proteins in the cell membrane that are required for the interaction with the outer media. We have sometimes flagella that is protruding out of the cell of the bacteria, and it's important for the movement of the bacteria. The flagella usually has a propulsion way of movement, and that's why it causes the bacteria to move. Not all bacteria have flagella, but there's a similarity between this bacteria and animal cells, like, for example, sperms. Sperms as well have flagella in which they can also move. We sometimes see a capsule, and the capsule, it's a cover-up of the bacterial cell. It also aids in the protection of the bacteria. And those types of bacteria that have capsules usually are hard to attack. Like not all antibiotics can attack the bacteria that has a capsule. So this is a general idea about the prokaryotic cell and how, what are the main components of the prokaryotic cell. We're going to move now to the second type of cells, which is the eukaryotic cells. When I say eukaryotic, eu means true, and karyot means that I have a true nucleus now. Here I'm talking about all other types of organisms, starting from protists to fungi to plants and then to animals. All these types of kingdoms, let's say, they are eukaryotes, and that means that they have a true nucleus and they have membrane-bound organelles. When we talked about the prokaryotes, as you can see here, there is no compartments. Everything is suspended inside the cytoplasm of the bacteria. So there's no need for compartmentalization, as we say, or the organelles are not enclosed into membranes, unlike the animals and plant cells that we're going to discuss. As you can see, the plant cell and the animal cell here, they have compartments. So all the organelles that they have inside are enclosed into membrane-bound organelles, we call it. Like look at the nucleus, the mitochondria. Here we have this is the mitochondria, for example. We have also the Golgi body. We have the centrioles. We have as well here chloroplasts in the plant cell. So generally, all the organelles are enclosed in membranes. Now, when we talk about the prokaryotes, we can see now the main components, as we have already discussed. We have the single chromosomes, which is the nucleoid or the DNA, the ribosomes for the proteins, the cell wall, and all the stuff that we talked about in the cell wall. The main component of the cell wall of the bacteria is the peptidoglycan. When we say peptidoglycan, it's a protein with a carbohydrate. Together, they form the bacterial cell wall. It's different, of course, from the other types of cell walls that are found in plant cells or even in fungi. Now, this is the capsule that we have already discussed, and the cytoplasm, of course, which is the fluid-like structure in which all the organelles are enclosed. Now, this is what I call the gram-positive and gram-negative bacteria. Now, according to the structure of the cell wall of the bacteria, scientists have decided to distinguish between those types of bacteria so that they can know which type of antibiotic should be used whenever we want to kill that bacteria. Now, a scientist called Gram, he had discovered a dye, and this dye, he used it on the bacteria. And this dye has to do with the peptidoglycan interaction that causes the color of the bacteria changes under the microscope. Now, as you can see, we have two different types of cell wall that covers the bacteria. The first one here, as you can see, the peptidoglycan layer is very thick. And this type of layer is going to interact with the stain of the scientist who is Gram, and that's why we call it gram-positive. So this gram-positive bacteria means that it has a thick layer of peptidoglycan, and the color that it shows is basically the blue color, which is the color of the stain. Now, this makes the gram-positive bacteria suitable for certain types of antibiotics to be used that can break down the peptidoglycan and easily can kill the bacteria. Now, the gram-negative bacteria, look at the structure of the cell wall. You can see how the peptidoglycan layer is very small, and it is actually enclosed between two layers of the membrane. So this would make it hard for the stain of Gram to interact with the peptidoglycan layer, and that's what causes the whole bacteria to become in another color, which is the red color. So eventually, when we talk about the gram-negative bacteria, now we know that the peptidoglycan layer is far away from being attacked by the antibiotic. So we have to use another type of antibiotic to attack other membranes of the bacteria so that we can make it effective against it. So here we can say that it's a little bit harder to kill rather than the gram-positive bacteria that is available here. Now, when we look at the basic structure of the plant cell and the animal cell, of course, we have already taken these types of organelles, but we have to go into deeper now, highlight about how the structures are related to their function and why they are important to study, because each type of cell may include the same organelles. However, sometimes we might have other organelles more than others because the function of the cell is actually different. Now, when we look generally at the plant cell or the animal cell, we have a lot in common between the animal cell and the plant cell. Like, for example, both they have ribosomes, both have the nucleus, the nucleolus, the mitochondria, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, the Golgi apparatus, the cytoplasm, the cell membrane. All of these are common. However, by looking at the plant cell, we can see that plant cells have chloroplasts, of course, for the photosynthesis. They have the cell wall that has cellulose, and they have, of course, the large vacuole, which is the permanent vacuole, we call it, and it's very obvious whenever we look into the plant cells. However, in the animal cells, we can see that we have the centrioles and we have the lysosomes as well. That is common only in animal cells. And we have also some of the microtubules that are responsible for the spindle fibers that are responsible for the cell division during the mitosis and the meiosis cell divisions. Now, by looking at the eukaryotic cells in general, we will be talking about the plasma cell membrane in more detailing. Then we're going to talk about the membrane-bound organelles, which is all the organelles that are available inside the cell. And of course, we will talk about the cytoplasm, which is the fluid-like structure, or the cytosol. When I say cytoplasm, I'm talking about the fluid structure itself. And when I say cytosol, it's the fluid structure along with the organelles that are available inside that cell. So let's start now with the first organelle, which is the most important organelle in the eukaryotes, which is the nucleus. Now, by looking at the nucleus, this is an enlarged, of course, picture of the nucleus. You can see how the nucleus is surrounded by a nuclear envelope, we call it, or a nuclear membrane. So it's one of the membrane-bound organelles. It has a membrane that is surrounding it, and these membranes are very important because we need to put things in and out of the cell, and this membrane is basically resembles the plasma membrane of the whole cell as a whole. The nuclear envelope of the nucleus has nuclear pores, and these pores are small openings in which we can release the ribosomes out, because the ribosomes are made inside the nucleolus of the nucleus, and then they are released outside into the cytoplasm. And one more thing that has to be also going out of the pores, it's the mRNA during the protein synthesis that takes place on the ribosomes as well. So we have now the nuclear envelope in which we have nuclear pores, and then we have the chromatin. As you can see, the chromatin is basically the DNA structure that is tangled along with the proteins that is also available in histones, we call them. And these proteins are responsible for the coiling of the DNA whenever we want to form chromosomes. So when the cell is not dividing, then the DNA would be as a chromatin structure, which is a thread-like structure. And we have proteins in between that are called histones, and this is what makes the whole chromatin. By looking at the nucleolus is a smaller thing of the nucleus, which is also a dense part of the nucleus that contains DNA, proteins, and as well RNA. The RNA is very important here because it is responsible also for the production of the ribosomes. So this is the function of the nucleolus. All right. Now, by looking at the DNA chromatin, this is a picture of the DNA. As we said, the chromatin is a DNA along with the proteins, and whenever they are coiled, they are called the chromosomes. Let's move on now to the second organelle, which is the ribosome. Look at the ribosome. It looks like a cartoon shape, where we have two subunits that are attached together. These two subunits, they are very tiny and very small, and they are attached to the rough endoplasmic reticulum whenever they want to make proteins. So the main function of the ribosome is basically the protein synthesis, and they are found just out of the nucleus directly on the rough endoplasmic reticulum. The ribosomes are made of small subunit and large subunit, and the most important thing is that they associate themselves together with the mRNA, which is carrying the codons from the DNA whenever we want to make protein synthesis. We're going to discuss this, of course, whenever we talk about the molecular genetics. Now, the endoplasmic reticulum, which is part that is attached to the outside of the nucleus, and the endoplasmic reticulum has a structure that is very nice, which is like folded, we call it folded membranes. By looking at these endoplasmic reticulum, we can see that we have two types. We have the rough endoplasmic reticulum, and we have the smooth endoplasmic reticulum. This is called the Golgi body, and we will discuss it after we're done with the rough and smooth. So we have two types of the rough endoplasmic reticulum. The rough endoplasmic reticulum is called rough because it has ribosomes. And so the function of the rough endoplasmic reticulum is basically about production of proteins because it is covered with all the ribosomes in here. Now for the smooth endoplasmic reticulum, it's also folded membranes, and the smooth endoplasmic reticulum is responsible for the production of fats and lipids that are needed in our body, like cholesterol, for example, like steroids, like waxes. All of these are steroids and triglycerides that are needed in our body, so they are produced in the smooth endoplasmic reticulum. And as well, the smooth endoplasmic reticulum may also be responsible for the detoxification and the breaking down of some of the carbohydrates that we do not need. Now, by looking at these two, the rough and the smooth endoplasmic reticulum, they are very close to each other in the cell. So by looking at the nucleus, for example, here, we can see how the rough and then the smooth endoplasmic reticulum are very close. Because once the mRNA is released from the nucleus, it will now enter into the process of protein synthesis on the rough endoplasmic reticulum. Now, the process by which we are moving these molecules, which is the protein that is produced eventually, it is through vesicles. So the vesicles now are going to encounter the protein that is being produced, and then it will move across the membrane until it reaches the other organelle, which is very important, which is the Golgi apparatus. The Golgi apparatus here is folded membrane by which we are using it for the packaging of the molecules that are produced. It's also important for the modification of these proteins. And modification means that we are adding things to the protein. We're adding molecules to the protein. Like, for example, if we have a protein that is produced and we need to add carbohydrates to it so that we can make glycoproteins, this process is made inside the Golgi apparatus. And if you want to add, for example, lipids to the proteins that are being produced from the rough endoplasmic reticulum, then we are producing lipoprotein, which is basically the LDL and HDL cholesterol that is found inside our body. So the Golgi apparatus is receiving the proteins that are produced from the rough endoplasmic reticulum through vesicles. These vesicles are transported into the Golgi apparatus, and then they will pass through the membranes inside where all the packaging, modification, and of course transport is taking place inside the Golgi. Once we reach the last part of the Golgi apparatus, where now the protein is ready to be excreted outside the cell or even to be transported inside the cell, then now the protein will be released as a vesicle, and we call it excretory vesicles because we need to excrete it outside the cell. Once this vesicle reaches the membrane of the cell as a whole, it will now make exocytosis. And exocytosis means that I am releasing my molecule outside the cell directly through the exocytosis process, where the membrane of the vesicle is now going to interact with the phospholipids of the membrane of the whole cell as a whole. So in general now, the rough endoplasmic reticulum is responsible for the protein synthesis because it has ribosomes. Once the proteins are produced, they are encountered into vesicles, and these vesicles are going to move through the Golgi apparatus, where they're going to be packed, they're going to be modified, and they're going to be transported to the outer area of the cell. So by exocytosis now, the excretory vesicles are going to include themselves with the membrane of the cell, and they will be released outside the cell. Like, for example, if I need, let's say, insulin. Insulin is a protein produced inside the rough endoplasmic reticulum. Then it is packed inside my Golgi body, and then it is released outside the beta cells of the islets of Langerhans of pancreas. So if this was the beta cell of the pancreas, then the insulin is going to be released by exocytosis outside to the body, to the blood, where it will run and do its job. If I'm talking about an enzyme, for example, that is used inside the cell, like let's say DNA polymerase or ligase, which is inside the nucleus, then the enzyme is going to be released by a vesicle into the cytoplasm and then taken up by the nucleus again. And as for the smooth endoplasmic reticulum, we said it has two functions, either to produce lipids or to break down carbohydrates later. This is basically the smooth endoplasmic reticulum. And if we're talking about the function, which is the production of lipids, that means it has to be in high amounts in the cells that are producing a lot of lipids. Like, for example, the testes and the ovaries, they are producing the hormones, testosterone, estrogen, and progesterone, for example, and these are steroids. So they have to be in high amounts in these types of cells because we are producing these types of lipids. This is the rough endoplasmic reticulum, and as I said, we have ribosomes. It's filled with ribosomes, where it is responsible for the production of protein and the folding, of course, of protein in the 3D structure. Remember when we talked about the proteins, how they have primary, secondary, tertiary, and quaternary structure? Now, once the primary structure is produced in the ribosomes, in the rough endoplasmic reticulum, they might fold it to make it into the tertiary structure, for example, or even the quaternary, so that it can be released also into the Golgi apparatus. This is how the Golgi apparatus is working. And if we're talking about the sequence of events, it's the rough endoplasmic reticulum. The secretory vesicle that is released from the rough endoplasmic reticulum is going to reach the Golgi, where now it's going to be modified, folded, transported, packed, and then through the secretory vesicle, it's going to leave through the membrane for the release outside the cell. Now let's move on to the next, which is the mitochondria. And the mitochondria is a very, very important organelle inside the cells of the eukaryotes, whether we're talking about protists, fungus, animals, or plants, because the mitochondria is the powerhouse of the cell. It is responsible for all the energy produced inside the cell. None of the cells in our body can function without energy. So we need energy to be produced through this mitochondria. The mitochondria is basically made of two layers of membranes. It has the outer membrane and it has an inner folded membrane, as you can see. The inner folded membrane, it is folded because we need to maximize the surface area for the reactions that are taking place inside the mitochondria. Now this folded membrane is called the crista. And on the crista, we have a lot of chemical reactions that are taking place, by which the energy is produced at most in the on the crista by the electron transport chain and the series of events that are taking place through the membranes of the crista. Now, for the ATP synthesis, we need an enzyme that is called ATP synthase, and this enzyme is found a lot on the crista membrane, where the production of ATP is at its maximum. The mitochondria has its own ribosomes to produce its own proteins, its own enzymes, and as well the mitochondria has its own DNA. Now, the DNA that is found inside the mitochondria is also responsible for its replication. So when we talk about the mitochondria that is needed in muscle cells, for example, we need to produce more mitochondria because the muscle cells are consuming a lot of energy. At this point, the mitochondria can replicate itself easily, and because it has its own DNA. So this is how the mitochondria is working by and the membranes that it has, where the crista is responsible for the electron transport chain that takes place after the series of events that are responsible for the respiration process and the production of energy. Let's look now at the lysosomes. And the lysosomes, the word lyse means that we are breaking down. So they are responsible basically for the breaking down of any unwanted material inside the cell. Now, when we talk about unwanted material, it's like a pathogen, for example, that has entered. And that's why we find a lot of lysosomes inside white blood cells, because they are fighting any infection that is taking place inside our body. So lysosomes basically have digestive enzymes, hydrolytic enzymes, in which they can break down any unwanted material, any wasted material. And at some point, when the cell dies and it's no longer of use, now we need to make death of that cell. So the biggest issue now here is about the lysosomes, that they take in all the materials of the cell, they enclose it and they digest it and then recycle all the materials to be used for next building up of cells. The word death of cell is called apoptosis. And the apoptosis is very important because it's a programmed cell death. Whenever we need to get rid of that cell, then we take it in the lysosome so that we can get rid of all the materials and recycle them to be used again. Let's talk about the vacuoles or the vesicles that are also found inside the animal cells and the plant cells. Now, if we talk about the plant cells have large vacuoles, and they are very obvious because the vacuoles are the area where we store water and minerals. We might store some dyes. We might store toxins that are available in certain types of plants. So it's a storage area, and the membrane that surrounds the vacuole is called the tonoplast. It's a large vacuole to keep all the water and minerals in, so that we can function and make photosynthesis whenever we need. The vacuole is also very important because it maintains the shape of the plant cell. So whenever we lack a lot of water in a plant cell, the plant cell remains intact because we have a cell wall, and the shrinkage of the vacuole will not affect that much the cell wall. Even if we have a lot of water inside the vacuole, it will also become turgid, and it will make the plant cell more rigid and more firm, so that it can withstand any, let's say, wind or any attack from the outside. Now, sometimes, and some animals like the protists, for example, like the amoeba, they have a contractile vacuole in which they can pump water outside their body whenever they are filled with it. Because if they live, for example, in an area where we have high water content, like in freshwater ponds, for example, we can see that how the water is entering into these types of cells into the vacuole that they have. So what will they do? They're going to contract those vacuoles and expel the water outside, so that they won't burst and be filled with that amount of water. Let's move on now to the chloroplast. And the chloroplasts as well, they look like the mitochondria. They're oval in shape, and they have also a double membrane. The membrane that is outside is called the outer membrane. And then we have the inner membrane, which has all the discs, as you can see. These discs are called thylakoid membranes, and these thylakoid membranes make a group of discs together that are called the granum. We have a lot of granum, and the granum are responsible for the photosynthesis, especially when we talk about step number 1 in photosynthesis. And then we have the area which is called the stroma, and the stroma is the area in between the granum, where also we have the second step of photosynthesis taking place at that point. Now it's time for our question. We're done now with the first part of this chapter, and we'll continue now with the second part. Thank you.